Gregor Mendel is known as the father of genetics. He was an Augustinian monk and ordained as a priest. A graduate of the University of Vienna, Mendel was trained in physics and mathematics. In 1856, Mendel began to study pea plants, analyzing thousands over 8 years in his monastery garden. His findings were instrumental in providing the framework for understanding inheritance patterns. The main reasons Mendel chose to study inheritance with the garden pea is that they were available in many varieties that differed in characteristics, such as flower color, flower position, seed shape, pod shape, and height. Garden peas are also self-fertilizing and easy for breeding true breeding lineages. Further, the flowers of garden peas are large and easy to manually fertilize. Mendel's laws are the law of segregation and the law of independent assortment. To understand these laws, it is important to understand the following concepts and terms. A character is a general feature of an organism. A trait is an identifiable characteristic of an organism. Each of the characters of pea plants that Mendel followed were found in two discrete variants, which were traits. For example, seed shape is a character and round and wrinkled are traits. A gene is a unit of heredity that is transferred from parent to offspring, contributing to the characteristics or traits of an organism. An allele is a variant form of a gene. For example, a tall allele would be a variant form of the gene for height. An organism with two of the same alleles of a gene is homozygous, and an organism with two different alleles of a gene is heterozygous. A phenotype is the visual characteristics of an organism that are the result of the expression of its genes. A genotype is the genetic composition of the organism. A dominant trait is a trait that is the displayed trait. A recessive trait is a trait that is not expressed in the presence of a dominant trait. Mendel discovered the law of segregation using a monohybrid cross. A monohybrid cross is the cross that observes the inheritance pattern in variants of only one character. To explain the monohybrid cross, let's examine one of Mendel's characters, such as seed color. The two traits for seed color are yellow and green. The yellow trait is the dominant trait, shown by two uppercase Y alleles, and the green trait is the recessive trait, shown by two lowercase y alleles. What happens when one true breeding organism breeds with another true breeding organism that has a different trait? To see the inheritance pattern in the monohybrid cross for seed color, it's helpful to use a Punnett square. The Punnett square allows us to determine the different genotypes of the offspring pea plants. Each parent pea plant will contribute one of their two alleles to the offspring pea plant. To make a Punnett square, we write down the genotypes of both parent pea plants and the possible gametes that each parent can make. Next, we create an empty Punnett square with 4 boxes. The number of columns equals the number of male gametes, and the number of rows equals the number of female gametes. Next, we fill in the possible genotypes by combining corresponding alleles. Lastly, we find the proportions of genotypes and phenotypes of the offspring. The genotypes are shown in the boxes of the Punnett square, and the phenotypes are determined by the dominant allele. We then make another cross and Punnett square with 2 offspring in the F1 generation that self-fertilize to produce 3 yellow seed offspring and 1 green seed offspring. These offspring represent the F2 generation. Seen in the monohybrid cross, the 2 alleles of a gene separate during gamete formation so that each sperm and egg receives only 1 allele. The F2 generation from the original monohybrid cross of 2 true breeding organisms with different traits will always produce a 3:1 phenotypic ratio and a 1:1 genotypic ratio. Mendel continued his work on inheritance patterns by conducting crosses that followed inheritance of 2 different characters. This type of cross is known as the dihybrid cross. Taking another one of Mendel's characters for the dihybrid cross, let's use seed shape with seed color. The true breeding plant of one strain could have yellow and round seeds, and the true breeding plant of another strain could have green and wrinkled seeds. In this scenario, yellow seed color and round seed shape are the dominant traits. Mendel had 2 hypotheses for the assortment of 2 different genes. One hypothesis was linked assortment. Linked assortment is where the 2 genes are linked, so variants that occur together in the parents are inherited by the offspring as a unit. In this example, the yellow seed allele, uppercase Y, would be inherited with the allele for round seeds, uppercase R, and the alleles for green seeds, lowercase y, would be inherited with the allele for wrinkled seeds, lowercase r. There would be 2 possible male gametes and 2 possible female gametes. The other hypothesis is independent assortment. In independent assortment, the 2 genes are independent of one another. Their alleles randomly distribute into gametes. In a dihybrid cross, there would be 4 possible male gametes and 4 possible female gametes. If linked assortment is correct, the F1 plants can produce only uppercase YR and lowercase yr gametes. In the F2 generation, the gametes would combine to produce 1 homozygous yellow and round seed, 2 heterozygous yellow and round seeds, and 1 homozygous green and wrinkled seed. The ratio of phenotypes would be 3 yellow and round to 1 green and wrinkled. If independent assortment is correct, the F1 plants can produce 4 gametes, uppercase YR, uppercase Y and lowercase y and uppercase r, and lowercase yr. Therefore, the F2 generation includes yellow round seeds, yellow wrinkled seeds, green round seeds, and green wrinkled seeds. The ratio of phenotypes would be 9:3:3:1. Due to the dominant traits yellow and round, Mendel observed that the F1 dihybrids were all yellow and round seeds. This is true for each hypothesis. Yet, the F2 generation observations proved independent assortment because Mendel observed 4 phenotypically different types of offspring. Therefore, the law of independent assortment is correct. This concludes an overview on Mendel's laws, which profoundly impacted later research on chromosomes and the processes of mitosis and meiosis.